So I'm happy to introduce James Hawley to you today. James obtained a Bachelor of Mathematics at the University of Waterloo in Ontario. He did so very successfully, making the Dean's Honours list, for example, getting more distinctions as well. And during his undergrad, James already gained some research experience, including during one project on statistical analysis of prostate cancer data in the laboratory of Dr. Paul Buttress. So this must have raised an interest because in 2016, he started his PhD research at the University of Toronto in the lab of Mathieu Lupien at Princess Margaret Hospital. And there he's been using computational biology to better understand cancer epigenetics, particularly in prostate cancer. Many organizations have already recognized his potential by awarding him scholarships, such as the Merit Entrance Scholarship at the start of his PhD, and more recently, the Frederick Banting and Charles Best Canada Graduate Scholarship. So I'm very excited to hear what you've been working on, James, and please go ahead.
All right, well, thank you for the introduction, and thanks to the organizers for having me speak here. So I'm going to be talking about some recent work that I and one of the former PhD students in my lab, Stanley Zhao, have been working on. This is a preprint that we put up on bioRxiv earlier this year, but it focuses on the cis-regulatory interactions and topological changes in prostate cancer. So with that, I'll get started. So, I don't need to explain that much to this group here about the structure of chromatin, but I'll just kind of cover the basics. We know that DNA is not just randomly floating around inside the cell nucleus. It has this structure at multiple levels, starting from the very smallest point where the DNA is wrapped around nucleosomes to these more focal interactions between different DNA elements, between enhancers and promoters or insulators, up to these hubs of chromatin interactions, TADs and compartments, and then all the way up to chromosomal territories and how they isolate inside the nucleus. And so this organization is important for gene regulation, and disruptions to it can raise some problems. This has been increasingly recognized over the last few years. There have been a couple of very interesting papers that have discussed how alterations to this organization, ectopic interactions with enhancers and genes can lead to their overexpression. And this has been found in a number of developmental diseases as well as cancers. But how different components of the genetic architecture, the genetics, the organization of the chromatin and chromatin states, how all of these things cooperate together isn't entirely clear in general. There's still lots that needs to be figured out at the moment. If you were here a couple of weeks ago, you would have seen a good presentation by Dr. Furlong discussing some recent work about the decoupling between chromatin organization and gene expression changes in Drosophila, and how even though you have many of these structural variants, these balanced duplicate or rearrangements, they don't necessarily lead to widespread changes in gene expression. And so, how SVs, structural variants, can disrupt this organization in disease is still a bit of an open question. And so my lab specifically is focused on how all of these things operate in the context of cancer. And so one model that we really wanted to look at that we thought would be a good place to study this relationship was in prostate cancer. More than 95% of prostate tumors harbor structural variants. These cover important oncogenes like PTEN, MYC, and ERG. And we know that structural variants play an important role in the progression of this disease as well. Some recent work has been able to highlight the amplification of an androgen receptor itself, or an upstream enhancer of the androgen receptor, leads to its overexpression in metastatic prostate cancer, a more lethal version of the disease. And similarly, amplifications of enhancers to the FOXA1 transcription factor are also overrepresented in metastatic disease compared to different types of structural variants. And so we know that structural variants are a very important component of how prostate cancer develops and progresses in response to treatment or more aggressive disease generally. And so in order to investigate this, I'll briefly summarize a lot of work that went into this. What we were able to do is we optimized a low input Hi-C protocol to work with these primary tissue slides. One issue that we've seen in the field is that the study of chromatin organization often happens in the context of cell lines or model organisms, and very little has been discovered so far about how chromatin is organized from primary patient tissues themselves. And so we were able to optimize this particular protocol to work on 10 micron sections on tissue slides. And we were able to do this for 12 primary prostate tumor specimens and 5 benign prostate specimens as well. And so we were able to incorporate our chromatin organization data with other multi-omic data for these matched samples that have been previously published as a part of the CPC Gene Prostate Cancer project. And so with that, we thought that this was a good context for studying all of these sort of complex relationships, how structural variants interact with chromatin organization, how chromatin organization changes just over oncogenesis itself, and how these things have worked in tandem with each other in disease. And so the first thing that we really wanted to look at was, how does higher order chromatin organization change over oncogenesis itself? We looked at the topologically associated domains, and we found that regardless of what sort of magnification we looked at, whether these, biggest domain TADs or the sub-TADs contained within them, regardless of that size, we found that they were largely similar between the tumor and benign tissue samples. And so we didn't really see any sort of evidence of large-scale, higher-order changes to the chromatin over oncogenesis. Similarly, we found this result in compartments as well. There were a couple recurrent compartmental changes, but these weren't associated with changes to, say, gene expression. So overall, we found this higher order chromatin organization seems to be pretty stable over oncogenesis. The result is a little bit, let's say, muddier, but largely similar for these more focal interactions as well. We were able to identify focal chromatin interactions across all of our primary tissue samples. And what we found is that the interactions that were, say, only identified in tumors were similarly enriched for contacts in both the tumor samples and the benigns, and vice versa, that interactions that were only detected in benign samples were similarly enriched in the tumor samples themselves. And so while we were able to find these sort of subtle differences in some of the focal chromatin interactions, largely they were pretty consistent in that most of the focal chromatin interactions were actually shared between benign and tumor tissue. And so largely this sort of pointed us in the direction of genome topology is actually pretty stable across oncogenesis. We didn't find evidence of large scale rewiring of the chromatin, and this was just essentially largely stable. There are these subtle differences that may be contributing to differences in gene expression programs between benign and tumor tissue, but on the whole, it seemed to be pretty stable. And so this sort of led us to this question of, benign and tumor tissue, they're phenotypically different. What's actually going on here? Well, one thing that we actually know about these two different tissues is that they have these differences in structural variant abundance. And so using recent developments in the detection of structural variants, we were able to identify, structural variants across the genome in all of our tumor samples using both Hi-C and previously published whole genome sequencing data. And largely what we found is that Hi-C was able to detect largely many more interchromosomal breakpoints than whole genome sequencing was. And having the two of them together helped us sort of resolve some of these more complex events that we typically see in prostate cancer. But to largely, get back to the bigger question here, how do structural variants play into these differences between benign and tumor tissue? We wanted to look at how intra-TAD contacts, TADs themselves, were they disrupted by these structural variants or not? And largely what we found is that, not that much. Only a minority of TADs that contain structural variant breakpoints appeared to be altered in patient samples that had these structural variants compared to the patients that didn't. Most of them appeared to be not that altered. However, when we look at gene expression differences, what we find is that many structural variant breakpoints have associated changes in gene expression. But where genes are changing their expression is not necessarily where the TADs are being altered themselves. And so we find this sort of complex phenomenon or observation here that the coupling between TAD changes and gene expression changes, it's not necessarily that strong. That SVs can alter gene expression independently of intra-TAD contacts. We have both examples of when structural variants will change the composition of TAD interactions, but it's not associated with the changes in gene expression, and vice versa, where there's no apparent change to the TAD contacts, but we see large changes in gene expression. And so these things are, they appear to be somewhat independent of each other. And so, this really leads us to this question of how is this happening? What's actually going on as a result of these structural variants? How are they changing gene expression, either in the presence of or in the absence of changes to a higher order chromatin organization changes? And so we look for, sort of the recurrence of where these structural variant breakpoints were coming up. If we look at the location of the structural variant breakpoints and how they overlap active regulatory elements in prostate tissue. We looked at H3K27 acetylation peaks, and using some permutation tests, we found that the structural variant breakpoints are enriched in our active regulatory elements. This is a similar observation to what has been made using point mutations in prostate cancer, but we find that the structural variants themselves, their breakpoints appear to be enriched in these active regulatory elements. And so it suggests to us that SVs that change gene expression, they may be functioning by targeting the regulatory elements themselves. I mentioned a couple case examples in the introduction here, but we're finding this as a more sort of general pattern in prostate cancer SVs as well. And so when we focus on the structural variants that do change gene expression, and we can classify them by genes being only overexpressed, only underexpressed, or having a mixture of both over and underexpression, what we find is that most of the structural variants that change gene expression, they affect multiple genes, as you can see by each individual row containing multiple red dots. But moreover, we also find that most of these structural variants that affect gene expression change genes to both overexpress some and underexpress others. And so this coupling of SVs being enriched in active regulatory elements and the multiple directions in which genes expression is being changed led us to the suggestion that it seems to be that structural variants, when they do cause changes in gene expression, it may be through targeting active regulatory elements here. There is some work published a number of years ago about cis-regulatory element hijacking. How structural variants can either break TAD boundaries or move elements closer to genes, and that this new contact with active regulatory elements can lead to the overexpression of certain genes. And so this seems to be sort of the more sort of consistent idea here with what we've been able to find, that these altered regulatory element contacts may be what's leading to some of these genes being differentially expressed. And just for the sake of time, I'll really just sort of focus on one particular example here. We're able to look at the BRAF gene that is a part of an interchromosomal translocation between chromosome 7 and 19. And when we look at the Hi-C data and perform this virtual 4C analysis, what we find is that the point where the 3' end of BRAF is involved in this structural variant, it sort of loses contact with its original promoter. But what it does is that it joins to this new part on chromosome 19 and has these focal increases in interactions with these active regulatory elements on chromosome 19. And this BRAF transcript associated with this translocation is vastly overexpressed, about 5 times overexpressed compared to what it is in the non-mutant patients here. Now we have a couple more examples here, but I think for the purposes of time, I'll just really leave it at that. It brings us to this last conclusion that it appears that the structural variants repeatedly hijack cis-regulatory elements, and this may be what's causing the altered gene expression at structural variant antipodes. Largely, this doesn't appear to be a function of just general genome topological changes between benign and tumor tissue, but that it's more sort of a function of these focal interactions being rewired as a result of these genetic alterations. And so, to summarize what we've been able to do here in our work, we've optimized a low input Hi-C method for tissue slides so that we can better study primary tumor tissue instead of just cell lines or other model organisms. We found that chromatin organization is stable over prostate oncogenesis, is largely resistant to mutations, but that these focal interactions appear to be changing how genes may be expressed. And the broader conclusion of our work is that we've been able to develop a framework for investigating genetic alterations to this architecture using chromatin organization data to interpret and understand how these things may be working. So largely, I have a number of people to thank for this work, including my co-lead on this, Stanley Zhao, Kip, our lab tech, who is really instrumental for optimizing the Hi-C data, and other members of our lab and collaborators. And with that, I'm happy to take any questions.
Thank you. That was a beautiful talk, James. Okay, I will. Let's see. I think there are some hands up. Yes. First up, I'll unmute Alan Underhill and let him ask his question.
So beautiful work, James. Have you been able to adapt this approach to looking at treatment samples to see how treatment actually selects for structural variants?
This is a very interesting idea. So, like I mentioned in the intro, sort of adaptation to things like castration treatment, androgen deprivation that's commonly used in prostate cancer is, a common phenomenon. We haven't been able to explore those particular samples yet, but I think that is, a really interesting avenue that we could take this work to see how those focal interactions change as a result of this, adaptation to this new environment or in response to therapeutics.
Exactly. Great. Thanks.
I was also wondering, in the cases where you see H3K27 acetylene on these structural variants, have you compared to some kind of healthy prostate tissue? Like, are these places that, like sequences that in the original context would have had H3K27 acetyl already, or is it after their rearrangement or duplication or whatever happened, they gained this active mark?
Yeah, so that is a good question. So what is a recurrent structural variant in prostate cancer is this TMPRSS2 ERG fusion. And let me just see if I have this particular slide here, not that one, but this, for example. And so what we see is in all of our prostate cancer patients that have this particular fusion in the top here, you can see this hyperacetylated promoter region of TMPRSS2, and this gets fused to this 3' end of ERG. But the 3' end of ERG is also has this histone hyperacetylation, whereas the non-mutated patients, they still have this hyperacetylated TMPRSS2 promoter, but not the hyperacetylated ERG 3' end. And so we've seen this in a couple different cases where the structural variant is also associated with changes to the histone modifications, either for the gene that's being expressed or with some of those regulatory elements right around the breakpoints. And so with this, it's sort of pointing towards the increased either activity or novel usage of these elements in response to regulating these genes that are differentially expressed.
Yeah, I think this also relates to a question from Gabe Zentner, with you showing like the different types of tumors. He's asking, are there differences in the structural variants detected with different driver rearrangements? That, i.e., does a tumor with a TMPRSS2 ERG rearrangement have regulatory alterations different than the TMPRSS2 ETV?
Yeah, so this goes to work beyond just what we've presented here. Largely, we're able to find differences in abundances of structural variants between the TMPRSS2 ERG positive tumors and the TMPRSS2 ERG negative tumors. And previous work from our lab was also able to identify differences in the H3K27 acetylation, both genome-wide, and how that related to different changes in gene expression programs between these different subtypes of tumors. So yes, we have been able to see some of those changes in response to different tumor subtypes, and a bit of a speculation here, but that doesn't seem to be, it wouldn't be surprising to find that in other tumor types as well, that there are these subtype differences in either these focal chromatin interactions or the cis-regulatory landscape generally.
We have time for one final question. And for this, I will let Stephen Kravitz unmute himself.
Great. Wonderful seminar. Just a quick question. I know that you put the tissues on a solid support. Have you ever tried any fresh tissue to see whether the contacts remain?
That is a good question. So when we collected our benign tissue samples, we had specifically asked for them to be cryofrozen to sort of match the collection criteria for the tumor samples that were collected at an earlier time. I'm trying to think of the experimentation that we did for the protocol optimization. I don't think that we used any fresh tissue samples specifically, but I may be missing or forgetting some information about that.
So then, similarly, then usually, at least in pathology, tissues samples are stored in paraffin embedded.
Yep.
Have you actually compared the to your cryopreserved versus paraffin embedded samples?
No, so we haven't looked at that specific comparison. We'd seen that there'd been, say, 1 or 2 other publications that were able to do Hi-C on paraffin embedded tissues. They used a lot more cells than what we had access to for our tissue slides, which is why we optimized it for that. But we haven't done a specific comparison between preservation method and the contacts from the similar tissues.
Right. I'm just wondering. I would expect the contacts to probably change somewhat. And that's my only question, really.
Yeah, it's possible. I don't know if that has been studied more widely. I haven't been able to find anything, but that would be interesting to see whether that preservation method affected how these contacts were detected, for sure.
Great. Thanks.
Thanks.
Great. Thank you so much again, James. That was a beautiful talk.